like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong-Lu Lin School of Medicine Healthy Longevity webinar, and thanks for joining us. I'm very lucky tonight because I have a longtime friend, Heinrich Jasper, who will be on giving us a talk about stem cells and aging. But before we do that, I want to do two things. One, remind you to use the Q&A function during the talk so you can ask questions, and we'll try to get to those later. And second of all, introduce an intern for the Center for Healthy Longevity, Li Lian, who's talking about the TORC1 complex, chromatin organization, and autophagy.
Thank you for the introduction. As mentioned, I'm Li En, an intern under the Center for Healthy Longevity's clinical team. Currently, I'm helping out with the aging biomarker study in Singaporeans and a systematic review on rapamycin. Today, I'll be sharing an interesting study on how rapamycin can affect the way DNA is stored and how this can positively influence longevity. This study is titled "A TOR-C1 Histone Axis Regulates Chromatin Organization and Non-Canonical Induction of Autophagy to Ameliorate Aging." And this study is being carried out by Rishan Liu and team. Other research studies have shown that expression levels of histone proteins play a key role during the aging process. Thus, this study aims to study the role of histone expression in longevity in multicellular organisms, as well as to study the effect of rapamycin on histone proteins. Just to give you some background, histone proteins are proteins that act like spools in the cell, which the cell uses to wind your average 2-meter DNA into one cell, keeping it tidy and regulated. For this study, expression of core histone proteins was studied in both rapamycin-treated and control flies and mice. In flies, expression of histone proteins H3 and H4 was significantly increased in the intestine. Looking at the graph shown in this slide, the increase in expression of H3 and H4 histone proteins was also sufficient to increase longevity. However, rapamycin treatment did not affect the expression of H3 and H4 protein in brain, muscle, and fat. Given that there are many physiological and functional similarities between the fly and the mammalian intestine, this study proceeded to look at the effect of rapamycin in mice intestine. As shown in the graphs in this slide, the expression of all the core histone proteins, H2A, H2B, H3, and H4, in the small intestine was significantly increased by rapamycin treatment. Next, the research team found the expression of core histones to be increased in response to rapamycin treatment through EIF3 activity regulated by the mTOR-C1 pathway. Increased histone expression mediated increased expression of transcripts of autophagy-related genes BCHS and DOR. And this can be seen in the graphs in this slide by comparing the heights of the gray and the red bar charts. So The red bar chart represents when rapamycin treatment is present, while the gray one represents when rapamycin treatment is absent. Lastly, age-related intestinal diseases are driven by both unregulated intestinal stem cells, division, and loss of homeostasis in enterocytes. Rapamycin treatment proves to be useful for intestinal health by activating autophagy and reducing the Ph3 positive cell number, an indicator for intestinal stem cells proliferation in the intestine. So as you can see in the graphs in this slide, rapamycin helps to reduce the Ph3 positive cell number as indicated by the lower height of the red bar chart as compared to the gray bar chart. In summary, rapamycin targets the mTOR-C1 pathway, which regulates EIF3 activity. EIF3 activity stimulates an increase in histone expression, which then affects chromatin organization to activate autophagy. Autophagy in turn promotes intestinal health and longevity. Finally, I would like to conclude with some of the implications of this study. This study shows that the mTOR-C1 histone axis is at least partially conserved in mammals. The results of the study can possibly be translated into research for longevity in humans by offering new directions for therapeutics, anti-aging interventions, and the development of more precise pharmacological treatments. Thank you for your kind attention, and I hope you have taken away something from my presentation.
So tonight we're lucky to have Heinrich Jasper giving us a talk on stem cells and aging. He got his PhD from the University of Heidelberg and the EMBL, and in 2003 moved to the University of Rochester, where he became a professor and stayed there until 2010. At that point, he moved to the Buck Institute for Research on Aging in California, where I was, became a professor and later chief scientific officer. We were very lucky to collaborate together, or at least I was lucky, and produce some really interesting findings on stem cells and aging. And it's been a pleasure to work with him for that time. In 2017, he moved to Genentech and has been a director there since that time, where he's continued his work on stem cells and aging. Henry's run a number of awards, and I think I won't go through all of them for the sake of time. Just to tell you that his title tonight is Stem Cell Aging and Rejuvenation. Thanks for joining us, Henry, and I'm looking forward to your talk.
Thank you very much, Brian, for the nice introduction, and thank you very much for having me presenting in this forum. I'm really excited to give an overview of the work we've done to try and understand how aging impacts regeneration and how what the mechanisms are that control the loss of regenerative capacity that we all experience as we age. When we talk about regeneration, we're actually primarily thinking about stem cells, tissue stem cells that really are critical to allow adult tissues to renew themselves and to repair themselves. One thing I always try to remind people of is that these stem cells can actually become quite problematic as we age, as we have to maintain the stem cell population as a population of cells that have proliferative capacity. They are mitotically active, but can also need to be controlled in order to prevent these cells from becoming transformed. And so as we age, this balance between prevention of transformation, prevention of actually cancer, and the maintenance of regenerative capacity is something that becomes very difficult. And it is believed in the field that the reason our regenerative capacity in many tissues declines is because anti-cancer mechanisms are engaged as we age that ultimately limit the capacity of stem cells to regenerate. So really, what we need to understand is how this balance between regenerative capacity and anti-cancer mechanisms really can be optimized so that we can maintain our regenerative capacity in tissues as long as possible without leading to increased cancer incidence. As we age, stem cells are responding to a variety of environmental stressors that will also impact their health and ultimately contribute to the decline in regenerative capacity. And so what we're interested in the lab is to understand how tissue homeostasis is maintained and to understand whether we can find ways of modulating stem cell function to improve tissue maintenance, potentially increase tissue health, increase the health span of an individual or an animal. And so when we talk about tissue stem cells, by and large, we're talking about cells that are maintained often in an inactive state in tissues, but they get activated in response to a tissue damage or to a stress. And when they become activated, they commonly self-renew, so they divide to generate a new stem cell, and then also generate a progenitor cell that goes on to differentiate into these daughter cells, which ultimately will be the cells that make that perform the functions of the tissue. Inactive stem cells are often maintained in a microenvironment, is also called the niche. And the niche plays important roles in controlling stem cell activity. But stem cell activity is controlled at various levels. So there are obviously cell autonomous regulators, which means control mechanisms within the stem cell that maintain their stemness, their multipotency, their proliferative capacity, and ultimately also their lineage specification. And these control mechanisms are often transcriptional control mechanisms, such as epigenetic control, but also metabolic or redox state controls that control the ability of these stem cells to be active or to be generating various cell types. The niche itself will provide often trophic factors that control the maintenance of stem cells, but also regulate whether stem cells are active or inactive. And then there are systemic control mechanisms like hormones or growth factors in circulation that will impinge on stem cell activity, stem cell differentiation, on lineage specification, and ultimately the generation of daughter cells as well. Now, as we age, all of these things change. There are age-related changes in epigenetic control, so the intrinsic control of stem cell function. There are age-related changes in the niche itself. There are age-related changes in systemic growth factors. And in order to really understand how aging then leads to a decline in regenerative capacity, we need to understand all of these things in combination. And we need to ideally understand how these things will ultimately function together to drive a regenerative decline in the organism itself. And for that, obviously, we need model organisms. And we in my lab have used Drosophila as a quite accessible model system in which we can perturb stem cell function genetically and ask questions about how specific genes are important for the response of stem cells to environmental changes, and ultimately how specific genes are influencing age-related decline as well. And we primarily focus on the Drosophila intestine as a model system to study this. I will give an overview about the work we've done and how this has led to a fairly deep understanding of age-related regenerative decline in this tissue, but that also serves as a model for vertebrate systems in which similar stem cell decline and regenerative decline have been observed. And then I'll give a brief overview on a recent study that we've done, where we've tried to understand whether it is possible to use epigenetic reprogramming, which is ultimately a way to make somatic cells more into stem cells, as a strategy to rejuvenate tissues. So the Drosophila intestine is a really accessible model system because it is lined by a fairly simple epithelium. And obviously the intestinal epithelium serves as a barrier to the outside world. And this epithelium is actually exposed to bacteria as well as to obviously nutrients, but also to toxins, and gets damaged quite often. And therefore the cells in this epithelium need to be replaced quite often. And what you're seeing in the main frame here is actually a cross-section through this epithelium, where you can see these large cells, which are marked by a blue asterisk. These are enterocytes, which make up the bulk of this epithelium. And these enterocytes perform all the functions that the epithelium need to perform, which is to resorb nutrients, to fight infections, to interact with the commensal microbiota, as well as to release enzymes and also actually release hormones to some degree. And so these enterocytes are replaced quite often by stem cells that sit on the basal side of this epithelium and are surrounded by visceral muscle tissue. And what you can see on the left-hand side is a fly that has been taken apart, and you can see that the intestine as a whole is compartmentalized into an acidic region, which is labeled in yellow here, and that's the gastric region, the stomach basically, and then an anterior midgut and a posterior midgut that are labeled in blue here. And all along this gastrointestinal tract, you will see a similar epithelium with some peculiarities in different areas where stem cells behave a little bit differently. So we've actually learned quite a lot at this point about the makeup of this epithelium and the function and regulation of stem cells in this tissue. And what you can see on the top here is a cartoon of this cross-section again, with enterocytes labeled as EC, but also other cell types, enteroendocrine cells labeled as EE here, as well as enteroblasts, which are labeled in EB. These are cells that are ultimately progenitor cells derived from the intestinal stem cells labeled as ISC here. And these enteroblasts, the EB cells, will differentiate into the more functional cells of the tissue, the enteroendocrine cells and enterocytes. You can see at the bottom picture here an image from the basal side of the epithelium in which stem cells have been labeled in green, and the other cells, these large cells with a blue nucleus, are actually enterocytes. And so what this illustrates is that we actually have genetic markers and ways of tracing intestinal stem cells really very carefully, and that will allow us to really address very specific questions about stem cell regulation and function in the living organism. And that's important because it allows us to really ask questions about specific influence of aging and environmental factors on the behavior and the function of these cells, and then dissect this behavior genetically. And so because of this ease of access, we and many other labs have, over the last 10 years or so, learned in a lot of detail how intestinal stem cells are regulated. And you don't need to really understand anything about these specific pathways that I'm listing here. What this image is just illustrating is that we've learned in a lot of detail how, for example, enterocytes on the left-hand side are influencing stem cell proliferation, how the muscle surrounding the intestine is influencing stem cell maintenance and proliferation, how hemocytes at the top, these are blood cells in the fly, also influence stem cell activity, and how even insulin-producing cells, these IPCs on the right-hand side, which are actually located in the fly brain, also influence intestinal stem cells. And so because of this deep understanding of how the local and systemic environment is influencing intestinal stem cell function, we've also been able to really dissect in detail how aging is ultimately contributing to a decline in epithelial function. And what you see here on the right-hand side is an old fly, actually the intestine of an old fly, 45-day-old, is more or less about 60 years old in humans. So you can see there that nice arrangement of intestinal stem cells, which in the 3-day-old you see nicely dispersed throughout the epithelium, is lost. And you can see that there are these clusters of green cells that have lost stem cell morphology and are actually disrupting the function of the epithelium. And you can see this also in cross sections again through the epithelium. What you see on the top here is the image you saw before, which is an image of a 2-day-old fly. And what you see at the bottom is an image of a 60-day-old fly. And you can see labeled in red, or with these red asterisks, you see cells accumulating on the basal side of the epithelium, disrupting the apicobasal structure of the epithelium. And that will ultimately lead to a dysfunction of the epithelium, which even leads to a loss of the barrier function of the epithelium. I mentioned that this epithelium really is the barrier to the outside world. And if you lose barrier function of the epithelium, that really means that bacteria can get into the animal and lead to systemic infections. And so because of these type of observations, and then the genetic dissection of this process, we've come to a relatively good understanding for how, as the animal ages, the intestine declines. And what you're seeing here is a cartoon just depicting our current understanding of how this process happens. What you're seeing is that in the gastric region, the yellow area, as the animal ages, there's an initial process called metaplasia, where the gastric epithelial cells are losing their identity and are becoming more similar to the posterior epithelial cells. And this leads then to a dysfunction in the ability of the gastric region to secrete acid. So the acidification of the gastric region is lost, and you end up with a completely blue gut, as you can see on the right-hand side. And that actually means, in this case, that bacteria that are usually only able to colonize the anterior midgut can now also colonize the posterior midgut, because this acid barrier is lost. And that leads to what we call commensal dysbiosis, which then causes an inflammation in the posterior gut that leads to this so-called epithelial dysplasia. And I'm showing you here a number of example pictures that just illustrate these processes. The dysplasia is really this phenotype where you see the expansion of these green cells and the disruption of the epithelial structure. And that ultimately then leads to a loss of barrier function. And what you can see on the on the right side, is a example of 2 flies, one that is labeled as a non-smurf, and the other one is a smurf because it's completely blue. These flies have been feeding on a blue food that usually does not penetrate the intestine in a young animal. But as the animal ages, there will be more and more flies that when they feed this food will end up being completely blue. And that is just an illustration of the loss of barrier function in this epithelium. Now, I mentioned that we understand the control of stem cell activity quite well at this point, and we've learned more and more about different signaling mechanisms, different genes, different pathways that actually drive these age-related phenotypes. So I'm showing JAK-STAT signaling here, FOXO relish signaling acting later on the, so JAK-STAT actually leading to the metaplasia, FOXO and relish leading to a commensal dysbiosis and the epithelial dysplasia, and J&K ultimately leading to overproliferation of stem cells that then leads to this decline in barrier function as well. Interestingly, for all of these, because in the fly we can control gene expression in a cell type and temporally controlled manner, we can perturb all of these pathways selectively and push back this process of age-related degeneration of the epithelium. And that ultimately led to lifespan extension in all of these cases. So that just illustrates that this decline in regenerative capacity, this decline in homeostasis of the epithelium, ultimately also shortens the lifespan of the animal. Now, over the last few years, we've tried to understand the regulation of stem cells and the impact of age regulation in more and more detail. And I'm just going to go through a few vignettes of findings we've made that show how important it is to really dissect the regulation of stem cells in a lot of detail to understand these processes better. And so the first image here is from a paper that is actually a collaboration with Brian Kennedy, where we've asked what happens if stem cells have to re-engage in regeneration over and over again as the animal ages, right? You can imagine that the gut gets damaged, the stem cells are activated and need to replace or regenerate the tissue. When that happens, a pathway called TOR is activated in order to allow these stem cells to divide. But TOR signaling also can drive stem cells into differentiation. And what this means is once they're pushed into differentiation, they are not stem cells anymore. And so you're losing stem cells over time in the tissue, or at least you lose functional stem cells. And what we could show here is that really this repeated activation of TOR every time the stem cell divides, as the animal ages, leads to a loss of stem cell numbers. And we could show that this doesn't only happen in the fly gut, which is depicted over on the top of the right, but it also happens in the mouse trachea epithelium, which happens to be quite similar to the fly gut epithelium, as well as in satellite cells, which are the muscle stem cells of the mouse. So it really seems to be a fairly conserved mechanism for the loss of stem cell numbers and stem cell function. And we could indeed show that in all of these conditions, if you reduce or limit the activation of TOR signaling by exposing the animal to rapamycin, which is a TOR inhibitor, that was sufficient to actually maintain stem cell numbers. Now, going back to the stem cells themselves, stem cells actually have two modes of dividing. One is a symmetric division where they generate two new stem cells, and in certain contexts you need this. And the other one is an asymmetric division where you generate one new stem cell and a differentiating daughter cell, and that's important for the regeneration process. And we showed that as the animal ages, due to elevated Jun kinase activity, JNK activity, the mechanism that controls symmetric versus asymmetric divisions, which is actually the orientation of the mitotic spindle, is disrupted. And then you will get more spindles that lead to a symmetric division in the aging context. And that then contributes to the dysplasia that I showed earlier. And so all of these studies are published, so you can look them up if you're interested. The other age-related change we have observed is that normal metabolic processes in stem cells are disrupted. And what we could find is that looking very carefully at metabolites in living stem cells through live microscopy, we could show that in young stem cells, when they get activated, there is an uptake of calcium into mitochondria that controls the electron transport chain in order to produce enough ATP for the stem cell to divide and for the daughter cells to grow. Now, in old stem cells, even though they're continuously dividing, they do not actually take up calcium into the mitochondria, but they have switched their metabolism to a different way of generating ATP, which is ultimately glycolytic metabolism. And what is the consequence of this is that you generate too much lactate, which also for the tissue microenvironment is a problem. Now, this was interesting because if you, I actually skipped this slide, but this is interesting because in a cancer context, a similar switch happens. So if you actually compare normally growing tissues, cancer tissues, what you see is that cancer cells have switched to this glycolytic metabolism because it's a faster way of generating ATP, but it's not very efficient. Now, in the fly intestine, we could show that indeed when you activate certain oncogenic proteins in stem cells, you will induce the same type of switch. So really cancerous stem cells have this glycolytic metabolism. And what our findings suggest is that just the process of aging already leads to these stem cells entering this kind of precancerous state when it comes to their metabolic function. Now, these cell intrinsic mechanisms in stem cells that contribute to the decline in tissue function ultimately will lead to the degeneration of the tissue, right? And I've shown you this progression of intestinal decline in the fly. Ultimately, this is all associated with an inflammatory condition, and that means that the tissue itself is secreting inflammatory cytokines, which then will impact other tissues in the animal, and that's something we've asked about in one of the more recent studies, where we found that the age-related inflammation of the intestine actually contributes to the decline in olfactory perception in an aging fly. And so olfactory perception is known to decline with age, both in humans as well as in flies. And you can see this here in humans in the center and in flies on the right. As you know, olfactory and other sensory perception also is impacted by bacterial infections or by viral infections, including COVID-19, of course. And it's so to understand how these kind of inflammatory processes will ultimately lead to a decline in olfactory perception, we performed a study to ask whether, first of all, inflammation in the gut might influence olfaction as the animal ages, and secondly, how this happens. What are the cytokines that are being released? And what we found is that these cytokines labeled UPD2 and UPD3 here on the cartoon are indeed released from the aging intestine, and they will impact the antennal lobes, the AL here, which are ultimately neuronal structures in which you will find neurons that express olfactory receptors. And what we found is that UPD3 and UPD2 are impacting the antennal lobe function by changing the metabolism of glial cells, neuronal support cells, that are very important as metabolic supporters of olfactory neurons. And so what you see on the top here is just the depiction of the decline in the preference index. This is the P and I on the y-axis here, and you can see that flies actually have a higher preference for putrescine, that's one of their attractive odors, or a minus 75 or so preference for octanol, which is an aversive odor for these flies. But you can also see that as the animal ages on the x-axis, when they are 28, 44, 60 days old, they will have lost this preference for the attractive odors or the aversion for the aversive odors. Now, this loss in attraction or in aversion is actually rescued when you change signaling in response to UPD2 and UPD3 in the glial cells, and that's what you see in these green in the green data points there. And if we actually eliminate the ligand from the old gut, so we eliminating UPD2 and UPD3, you can also rescue this loss of attraction and aversion. So we could really show that it's this connection between the gut and the brain which ultimately leads to the decline in olfactory sensitivity. This again was published last year, so I don't want to go into any more detail here. Suffice it to say that in the paper we've identified the mechanism by which the specific ligands, these UPD2 and UPD3 ligands, influence signaling and metabolism in the glia that then ultimately causes changes in the olfactory neurons. So stem cell decline leads to a decline in homeostasis of the epithelium, and that ultimately impacts other physiological functions as well. And as you all experience with age, we see this more broader, more systemic decline in many tissues. And so one question, of course, that emerges is, what can we do about it? Can we actually restore tissue function in a way to rejuvenate us, or to, at the very least, fight back age-related decline, or improve health span in our own tissues? And so we recently published a study where we collaborated with Juan Carlos Espisuave-Montes lab at the Salk Institute to ask whether reprogramming, epigenetic reprogramming, is sufficient to rejuvenate tissues in healthy aging animals, in this case in mice. And so the concept here is that cells can be reprogrammed to pluripotency, and this has famously been demonstrated by Shinya Yamanaka, who got the Nobel Prize for the identification of 4 transcription factors. Let's just call them OSKM. These 4 transcription factors were sufficient to take a differentiated cell, an adult cell, and reprogram it completely back to a pluripotent stem cell, which means stem cells that can really generate any germ layer and can ultimately generate a whole organism. And so what you're seeing in this cartoon is basically a depiction of the differentiation path of cells from pluripotency down to a differentiated state. The idea is that ultimately, epigenetically, these cells go down a predetermined path and that you usually cannot go back from. Now, with reprogramming, clearly you can go back, and you can go back to full pluripotency. The question is whether it is enough to use this type of approach and improve tissue function by just partially getting these cells towards a healthier or younger state, rather than to full pluripotency. And that's something that has been actually demonstrated in progeria models by Espisuave-Montes lab in 2016. That's what you see there in, where it says rejuvenation, where they have shown that they can express these factors in the whole animal for a short period of time, and that is sufficient to bring these progeria animals back to a healthier state and even extend their lifespan. But it's also been shown by other labs. For example, Tom Waddell's lab in 2017 showed that you can take airway cells out of an animal, partially reprogram them in vitro, and then put them back into the animal where they can engraft and repair the lung in a bleomycin fibrosis model, for example. It's also been shown, again by Espisuave-Montes lab in 2018, that you can use transcription factors to reprogram epithelial cells or epidermal cells to treat cutaneous ulcers. And it was shown by David Sinclair's lab that you can express these 4 OSKM factors, or in this case, it was actually just 3 factors, OSK, in the retinal ganglion cells of the eye, and that would be sufficient to increase the repair of retinal ganglion cells in glaucoma models, where really retinal ganglion cells are being damaged, and they showed that you can improve repair of these cells using epigenetic reprogramming. And so, again, the concept here is that, and again, these images here are from Juan Carlos Espisuave-Montes' 2016 paper. The concept here is that these transcription factors, OCT4, SOX2, KLF4, and c-MYC, by being expressed for a short period of time, can actually reduce the damage or damage-associated epigenetic marks in cells. And that's what you see in these graphs. You can see that gamma H2AX, which is a marker for DNA damage, is reduced after treatment. P16 and IL-6 are reduced, P16 being a cell cycle inhibitor, which is a marker for senescence, and IL-6 being a cytokine, it is also a marker of senescence. The expression of these is reduced. And what you see on the right, in the right graph, is that the gamma H2AX mark is reduced, but then if you stop the expression of these factors, it comes back. And so what they showed at the time, and I'll go quickly through this slide, they showed at the time that this partial reprogramming protocol, which really in this case was 2 days of treatment where you express the factors and then you recover for 5 days, that you could use that protocol in whole animals to extend their lifespan, and also what you see on the right side to improve regeneration in the pancreas as well as in the muscle. And what we were asking in the current study was whether this would also be safe and effective in wild type, not in these progeria models, but in wild type animals. And we performed a study where we treated these animals for a long period of time using this protocol, 2 days on, 5 days off. And what we found is that compared to non-treated animals, there were no adverse effects as far as we could tell in terms of body weight, survival, as well as other parameters for health. But that if we looked at molecular markers for aging, transcriptional profiles, but also metabolic profiles, we could see that treatment with these 4 factors for 10 months or so, you do the 2 day on, 5 day off for 10 months, that would lead to a rejuvenation of a variety of tissues. And this rejuvenation could be seen also at the level of the serum metabolites, and it could be seen at the level of the methylation clock, which is a way to assess the biological age of a tissue. Importantly, in the skin, which was one of the tissues most affected by this, we could also show that this was sufficient to improve repair processes in the skin. So if you perform this 10 months of treatment and then you injure the animal, you induce a skin wound, the old animals that had been treated would ultimately close these wounds much faster than animals that had not been treated. So that really suggested that 4-factor reprogramming, partial reprogramming, is a viable way of systemically rejuvenating animals and ultimately improving tissue function. I'll leave you with a picture of the lab at Genentech at this point, and with acknowledgements, you can see the names here of people that we collaborated with. Thank you very much.
Thanks, Henry. That's great. I mean, it's nice to see where the research is going. I had a couple questions maybe about the partial reprogramming. So first of all, you found no incidence of tumors or any other issues in these animals that would suggest a risk?
As far as we could tell, no, there were no. The biggest concern you have with 4-factor reprogramming is ultimately teratomas, right? So that you would express these factors enough to get cells into full pluripotency, and once they become fully pluripotent, they will ultimately generate small embryos within the tissue, which is then ultimately a teratoma. We have not seen this, and the explanation for why we didn't see this is that really we had this 2-day treatment only. It's basically a very short-term expression of these factors, and then a recovery for 5 days before we treated them again, right? This is not to say, though, that in a human, for example, this might not happen after all. And so one has to take these things with a lot of caution, of course. At this point, we were surprised to see that the treatment for 7 to 10 months was actually safe as it was. I had not expected this, but it seems as if once you have established this type of kinetic, this type of protocol, you are indeed able to treat an animal for a long period of time and have a beneficial impact without the potential deleterious effects.
So are you doing survival studies on these mice?
Well, in this study, we had to take them down because we wanted to analyze their tissues. And so there is another cohort now where we're looking at longevity, but that will take a while.
Yeah, that's interesting. I wonder also, it might be interesting to induce this in an older animal, because one way of thinking about it is an active immune system may suppress cancer formation in any cell that happens to get too much of these factors in a young animal, but that may be more problematic later. So
Absolutely.
it would be interesting to start late in the mouse lifespan and see what happens too.
Yeah, and we actually, in the study, we did that. So we did and I didn't want to go into too much detail. We did various cohorts. We did cohorts where we started at 10 months of age and stopped at 22 months. But we also had cohorts where we started at 22 months of age and stopped at 25 months of age. And for these late onset, shorter term treated animals, we did not see much of a molecular rejuvenation, which suggests that this late onset treatment is not good enough anymore. We have crossed the threshold in which the tissues would be susceptible to improvement. But as far as I know, we also did not see increased tumor formation. So it was rather an ineffective treatment that we saw at that point.
Do you know that the factors were still getting expressed to the same level? I mean, there could be a...
Yeah, as far as we can tell, right. No, as far as we could tell, they were. But it is, of course, when you do these long-term or whole animal studies, disregard the possibility that you might have in certain tissues less expression, or that overall the kinetic was less effective as it used to be, right? Because you're only looking at the endpoint for transcriptional levels, and as you treat them, you're not constantly monitoring them. So there's certainly more studies to be done to be sure about this thing.
Well, it's very promising though. So
yeah,
hopefully
absolutely.
You'll get them done before I get too old to have the effects.
Right.
Speaking of, I mean, while we're on that, I mean, how far away do you think reprogramming is from being tested in human studies?
Well, I think the most likely scenario I see is that it will be used in very defined disease contexts. So not so much as an aging intervention, but for example, in the glaucoma context that David Sinclair has presented, where you are working within a very confined tissue, right, the eye, you can actually perform gene therapies, which is something that has been done in the eye, and where you can control the delivery very carefully. And so the eye in general is a perfect test case for these type of approaches. The other area I see this happening relatively soon may be the heart. There has been a study just last year showing that reprogramming in the heart in a similar way can actually reduce fibrosis in infarct models. Again, this was done in the mouse, so it will take some time to do this in humans. But these are the kind of scenarios where I believe there's an opportunity to move into a human context fairly quickly.
I would maybe just a couple of minutes on the intestine and the fly, and then we'll turn it over to the questions from the audience. It always struck me that, you know, you can just modify the stem cells in the intestine of the fly and get an effect on the aging of the whole organism. And I was always trying to go back and forth between the idea that the fly has very few stem cells in different tissues. And maybe that's giving undue importance to the intestine, or maybe there is something unique about the intestine. So would you think the same thing would happen in a mouse? If you could alter the intestinal stem cells, would that be sufficient to have effects on aging throughout the organism?
I think that's an interesting question. I think in the fly, I would argue that the intestinal epithelium clearly is one of the most limiting tissues, because as soon as it gets disrupted, you end up with systemic infections, right? And that really limits the lifespan of the animal. In the mouse, that might not be the case necessarily. However, once you lose barrier function in the epithelium of a mammal, you will have systemic effects as well, right? So you can imagine that conditions that allow you to improve on the barrier, that ensure sustained barrier function, will have a significant systemic impact, right? But obviously, vertebrates are more complex than flies, and many other tissues have evolved that may play a critical role as well. Certainly the cardiovascular system, if it fails, we're dead, right? So there's potentially other limiting tissues in vertebrates and in mammals that are equally important to be preserved, right?
That brings us to this link to olfaction, which I think is quite interesting, because as you probably are aware, that as people get older, getting them to eat normally is a big challenge. And one of the things elder people complain about is that their smell and taste is gone, and that could be related to declining olfaction. Do you think there's a intestinal link to olfactory changes in humans as well as they get older?
Well, we don't know. At this point, the link between the fly intestine and the olfactory system in the fly is really strong, and so it was actually really exciting to see this. I think in humans, the question will have to be first whether the loss of olfaction is indeed a consequence of these inflammatory processes, right? Mm-hmm. So we've identified a pathway that is conserved in vertebrates that controls and influences the glial cells and ultimately disrupts neurons. And so that would be the first thing to test, right, is whether one sees also an age-related change in juxtaphoreal signaling in glial cells that support olfactory neurons. If that exists, then it almost doesn't matter whether it's coming from the gut or from other sources. Yeah. You would have made a link between age-related inflammation and ultimately this loss of olfaction. And then there would be a way to target this as well.
Maybe what I'll do is bring Dong Lu in, and because we have some questions from the audience. Dong?
Thank you, Brian, and thanks very much, Prof Jasper, for the interesting talk. Now I'm going to start with some general questions from the audience first. It seems like many of our audience are interested in the fruit fly Drosophila. So we have a question from Charmaine first. She was asking, what are the major hallmarks of aging in this system, and how do these hallmarks differ from human beings?
So the molecular hallmarks of aging don't differ necessarily so much. We see, for example, age-related changes in epigenetic marks, just like we see in vertebrates. We see, as I mentioned, age-related changes in stem cell function. We see metabolic changes. There's even been reports of cellular senescence being observed in flies as well. So the question is always whether these molecular changes that we observe in vertebrates and invertebrates are ultimately mediated by the same mechanisms. And by and large, we've been able to say yes, in most cases, that's the case. Right? Now, again, I mentioned already in my answer to Brian that vertebrates and mammals specifically are more complex than flies, and you have other tissues and other interactions that you have to take into account. But ultimately, the fundamental hallmarks of the aging process are quite conserved.
Thanks for the answer, Prof. Jasper. So early on, you and Brian were discussing about the intestines of flies and humans. A related question would be from Aim. She was asking how similar a human intestine to Drosophilus, because she was wondering if humans as a more advanced organism would process different cell composition or maybe repair and regeneration mechanisms in our intestines than the fruit fly intestines.
Yes, and that's indeed the case. So humans and mice, the intestine is more complex. If you look at the small intestine, for example, you have very specialized regenerative structures, these crypts, that will ultimately have stem cells at the base of the crypt where they are very protected, and they will continuously produce new cells, and so that the turnover of the intestinal epithelium is pretty fast. However, there are tissues in vertebrates and in mice and in humans that are more similar to the fly intestine, and the tracheal epithelium is one example. So what I haven't gone into too much detail there, but the stem cells in the fly are maintained usually in an inactive state and are only activated when the tissue gets damaged, right? In the tracheal epithelium of mouse and human, that's the same thing. So you have basal cells on the basal side of the epithelium that act as stem cells, but they're not continuously dividing. There are no crypt structures there either. But as soon as the epithelium is damaged, the stem cells, these basal stem cells, will become activated and generate new cells. And even the molecular pathways that control differentiation and proliferation in these two tissues, in the fly intestine and the human trachea, are conserved and are very similar. So it's really a question of, are there the types of epithelia that we're looking at in the fly intestine, are they representative for similar epithelia in vertebrates? And that's certainly the case, but it's not necessarily the intestine.
I see. So now I'm kind of going to the next topic, which is stem cell biology in relation to aging. A question from David is that, in what tissue or organ system do we really start to observe a significant decrease in stem cell number and function during aging?
So it has really been described specifically in the brain, and so neuronal stem cells in the mouse decline in number and function. In the intestine, in the crypt of the mammalian or the mouse intestine, it has also been described that these LGR5 positive stem cells, which are populating the basal side of the crypt, decline in numbers over time. In the actually in the tracheal epithelium that I mentioned, we also see a decline in numbers of these. So it's widespread, right? And one should also say, though, that in other tissues, for example, the bone marrow, the hematopoietic stem cells, they do not decline in numbers, but actually expand and ultimately lead, and aging leads to a change in their lineage specification. They become more myeloid biased versus lymphoid biased. And so depending on which tissue you look at, which stem cell system you look at, aging may have a different impact.
In your opinion, is depletion of stem cell number and function a cause or a consequence of aging?
I think ultimately it's a consequence, and it will ultimately be a response to environmental and systemic changes, as well as to these intrinsic changes in metabolism and epigenetic function of cells. But then it will cause tissue decline with age, right? So it is ultimately caused by molecular changes, but then will have a significant impact on the health of the tissue.
In order to combat this decline in the function and number of the stem cells, a related question to that would be, are there any natural and practical ways to maintain or increase stem cell number and function during aging?
So we believe that the studies we've done point to a number of potential ways of doing that. And these are interventions that would impact stem cell metabolism, so interventions that would impact specific stress signaling pathways, maybe interventions that affect pathways like TOR signaling, these nutrient sensing pathways. So there are a number of potential ways of improving the function of stem cells as we age. And certainly these are this is an area of interest for us as we're at Genentech trying to target processes that are influenced by stem cell function, right? And so I think there's a wide range of possibilities, but a lot of work needs to be done.
Thank you, Prof. Jasper. It seems like some of our audience are interested in OSTM. Mm-hmm. So a question from Shoubing would be, as mitochondrial dysfunction plays a role in cellular senescence, what is the role of OSKM in mitochondrial rejuvenation possibly?
That's an interesting question, and I don't know the answer to that. My expectation would be, since the stem cell epigenome is being rejuvenated, it seems likely that processes that have led to an, for example, proteome imbalance in mitochondria, where a lot of the proteins are encoded in the nuclear genome, will also be reversed. But it is also possible that some intrinsic mitochondrial DNA damages that may lead to a dysfunctional mitochondria may not be reversed, right? We have not looked into this in detail, but it is definitely a very interesting question to explore.
Thank you. We have more questions coming in. There's another question from Stephen. He was asking about an earlier study that earlier in one of your slides, you mentioned about mitochondrial function and improved efficiency of ATP production in stem cells. Do you also happen to observe increased mitophagy levels as well as in intestine stem cells? Multiple studies have shown that restoring mitophagy levels also improves stem cell function.
Yes, and we have not done this work, but other labs have shown that in the fly intestine, that's the same. That also happens. So you can, for example, improve stem cell function by overexpressing PGC1 alpha, for example, and really promoting mitochondrial biogenesis, as well as influencing Pink and Parkin to regulate mitophagy. These studies have been done. David Walker's lab, Lee and Jones' lab were very much involved in this, and have shown that perturbation of mitophagy can lead to an improvement in stem cell function and hence better health of the intestine and extended lifespan as well.
Maybe I can jump in really quick and ask a question, because I know a couple of people ask, you know, you've kind of switched from an academic career to a career in biotech industry. So we have a lot of young people watching. So maybe you can compare and contrast a little bit and the two options and talk about why you moved over to Genentech.
Yeah, sure. I have to say that the academic, I've always been an academic. I've been very interested in the fundamental questions of life sciences and to try and really understand just the mechanisms by which living organisms are controlled. And that's where academia excels, right? In academia, you have a lot of freedom to explore fundamental questions and really let your curiosity drive you. And I think that should not be dismissed as a driver of innovation as well. Now, industry, of course, has the mandate to try and develop effective interventions, especially healthcare industry. Effective interventions for human disease. And so you can imagine that your world gets a little bit more constrained because we're really focusing on trying to intervene in an effective way in a relatively short period of time and develop these type of intervention in a relatively short period of time. Now, as I developed my lab for 15 years, we were lucky enough to be able to explore a lot of fundamental questions that then eventually led me to ask, well, now that I've learned all these different things, wouldn't it be fun to try and really apply this knowledge and see, okay, now we've learned so many things about stem cells in the fly, but also in vertebrates. What if we can actually work in an industry where I can interact with really outstanding experts in drug development to try and perturb stem cells, improve their function, to try and really make an impact on human disease. And that's really what's exciting there. And so for for a young person thinking about their career, I think it's really something that one has to go and dig deep inside yourself and decide what is most interesting to you. I think both of these career paths have a lot of excitement. There's a lot of fun, really just solving problems, learning new things, trying to have an impact. And in academia, you have an impact on knowledge. In industry, you have an impact on patients. Both of these are really important. And both of these are really exciting careers. Now, as we look at biotech today, it's been exploding in terms of investments. There's really been an expansion of biotech and pharma over the last 15 years or so, which is really built on a lot of the fundamental knowledge that has been developed over the last 25 to 30 years, right? And so we're in this really exciting time right now where so much has been learned about living systems that really the sky's the limit at this point in biotech, right? And so it can be very, very exciting to be in this area and try to really apply these very different approaches, molecular approaches, physiologic approaches, antibody engineering, these kind of things to fundamental healthcare problems, right? So I would say, regardless of where you go in your career, life sciences in general is a very exciting field. And if you have an opportunity to combine these two things, and that's where I'm really lucky, is at Genentech specifically, there is an emphasis on basic research and on making this connection between basic research and drug development. And both sides of this coin really benefit from that. If you get that opportunity, you cannot say no. This is exciting.
Thank you a lot, Henry. That was a great answer, and it was wonderful having you on the show. We'll have you back again sometime. I want to remind people to use the chat function and the panelists and all attendees option to leave comments on the show. Also, look for news for our Center for Healthy Longevity and the School of Medicine after the end credits. We'll be back May 5th with a speaker to be determined. We're still scheduling, so stay tuned. It could be a surprise. And I want to leave you with a video of a 90-year-old Ironman.
I want to be 120. If you want to be 120, you got to get your ass out and go. It's that simple. You got to prepare for old age. Most people think that that's just something that happens, and it's not. What you do at 40 will determine what you can do at 80 or 90. I just wrote a 30-year mortgage. I sold a piece of property, and the guy wanted a 30-year mortgage, and I gave it to him, and I said, hey, you got an X on your back. He said, what do you mean? And I said, I said, I'm going to be here to collect that last payment. I'm almost 90 years old. I can jump out of that bed tomorrow morning and run up that hill without stopping. I don't hurt anywhere. I feel good. And I think that's the thing I'm most proud of.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm